this is going to be lab video number 7, and just a quick video on what to do for your unknown assignment. So there's going to be, you're either going to get assigned by, I'm going to email you whether you get unknown A or unknown B. So if you get unknown A, just ignore B. If you get B, ignore A. Right? These have results for 2 different bacteria. Okay, and you're supposed to identify what your bacteria is, and by looking at the results for different biochemical tests that are on these PowerPoint presentations, you're going to decide what your unknown is. So, for instance, this is a list of bacteria that your unknown can be. The first column tells you if it's bacillus or coccus. There's also a legend below here. Use the legend, it helps you decide what these mean. We're not going to do the spore, so don't worry about that. I give you results for the gram stain. So you're either going to see purple bacteria or pink bacteria. If they're purple, they're gram positive. If they're pink, they're gram negative. If they're round, you call them coccus. Right? If they're rod-shaped, you call them bacillus. Then there's it's called glucose fermentation, sucrose fermentation, lactose fermentation. Those 3 in that column are for the phenol red broth. And we're only using the lactose fermentation. Okay? There's not going to be a result for every biochemical test. There's not going to be a result for acid-fast stain, and there's not going to be a spore stain result either. So just ignore them. Or capsule, right? So ignore them. Ignore the phenol red broth for sucrose. The lactose I'll use. A means acid, made an acid, and it fermented lactose. If it didn't ferment lactose, you'd see a negative sign. The G means there's a bubble in the Durham tube. So it fermented, and you got a bubble. The starch hydrolysis on the starch plate, if it's positive or negative, right? If there's a halo, it's positive. If there isn't. So on that PowerPoint, you'll see a starch plate, and I think it says your unknown is organism A, right? So you look at the results that A gave, determine if it's positive or negative. If you go to the unknown worksheet, if you look on the lab PowerPoint that I gave you for, say you got unknown A, you would circle A up here. That's the one you're deciding what it is. And then on that PowerPoint, you would look at the negative stain. If you saw coccus, you might just put coccus here. If you see bacillus, put down bacillus. Right? If they're round, you have coccus. On the gram stain, is it gram positive or gram negative? Is it coccus or is it bacillus? Ignore the rest of these things. There's no results on either PowerPoint for them. Then when you get to the mannitol salt, on the PowerPoint there'll be a mannitol salt agar plate. And it'll be the results. You can use your lab PowerPoint to help you decide on the PowerPoint if it fermented mannitol or if it didn't ferment mannitol. Right? If there was no growth, then the bacteria didn't grow on the plate. So you would put that in your observations. Sometimes you don't really have a result. If there's no growth, there's no result. If it fermented mannitol, you might say that over here. Mannitol was fermented. Right? If it turned yellow, mannitol was fermented. Then if you get macaque, DSM methylene blue, if you got no growth, you put that there. If it grew, then you would describe what you saw. Starch plate. Did it ferment or did it break down starch, hydrolyzed starch, or didn't it? Okay, but you would look, the phenol red broth for lactose, right? There's a result for that broth on the PowerPoint, and you're to tell me, was lactose fermented or wasn't? What, did it make a gas or not? You know, what was in the Durham tube? In the SIP media, was indole produced? Was H2S gas produced? Was it motile or not? Yeah, actually, don't worry about the motility. Methyl red, post-scour. So sometimes it'll say the test was not completed. So you just ignore that. You can put that, write that down if you want. It's up to you. Okay. So you would use the biochemical data. I've got a spot on this biochemical data to put your results, if it's positive or negative for these tests. Now the MSA and McConkey, that's not so useful because they're more tests, but I put on there whether they grow or not for them. On MSA, only these 3 bacteria grow on it, so I put G, but I don't say anything about fermenting mannitol or anything like that on this. This is mainly used for all the different tests. The indole test, which is in the sim media, right? The H2S, if it turns black in the sim media, then you get an H2S result. Okay, so you might need to use your PowerPoint for the sim media to decide how you're going to answer whether it's positive or negative for the test. Because all that, the PowerPoints, you've been doing this for the last few weeks, right? In your principles, you've been explaining this. So the more time you spend on that, the better off you are. For the oxidase test, these are the ones that are positive. So you can put your unknown results at the bottom, then compare the results for your unknown, whether if you got unknown A or unknown B, they'll be different, the results, and then decide of all these bacteria, which bacteria is yours. And you can base it, sometimes only it takes one test to identify it. But these will help, it's sort of fun. Be sure when you write, now if you type it, if you use this form and type your genus and species, then you should italicize it. Okay? Now if you're going to do all this with a pen or pencil, and you write in your genus and species, be sure you underline it, even though there's an underline here. If I don't see a line under it, then I don't assume it's been underlined. Okay? So once you've completed this, you can save it and upload the folder to the assignment folder. Or you can take a picture of it if that's easier for you. But just go to assignments, click on the unknown assignment, and there's a folder there that you upload your file, and if you don't complete it by the due date, upload it anyway after it. I think you can still do that. I'll grade it. I may deduct points for being late. Upload the unknown worksheet. That's all you need. You don't need the data sheet, just the worksheet. Once you have identified, be sure you put a name there, and you need to let me know if it's A or B. I'll probably have a record of it because I email it to you. But just wait for my email. You'll have gotten my email by the time you see this. If you have any questions, let me know.